Okay, cool. Um, yeah, thanks for the invitation. I hope that the content of the talk is okay. I um, restructured it a bit based on feedback that I've received. So there's not so much intro to tissue clearing and how it works because I heard that you already had an intro to that. But of course, if there are questions about the method itself or how the tissue clearing works or some details on that, I'm more than happy to answer. Yeah, so maybe let's start with giving you some background on the group that I joined for my PhD. Um, basically, the group is, as was already said, led by Alad Robruck, and we are called the CBClab. The main focus of this group historically was actually on MRI imaging, and they had no connection to microscopy at all at first. They were doing a lot of diffusion MRI, both in vivo and ex vivo. And actually, the main focus of the group before I joined was to somehow investigate these more mesoscale features in the cortex of cortical architecture and cortical connectivity with MRI methods and kind of pushing the resolution of the MRI from the macro into the mesoscale. Uh, so Back in 2016, when I joined the lab, a lot of this kind of stuff was going on. So here in Maastricht, there are two very fancy MRI scanners, and I have to apologize. I don't know too much on the MRI side of the topic because that's not really my thing. But we have a 7 Tesla and a 9.4 Tesla scanners, so ultra high field scanners, and basically with them it's possible to generate MRI images with much higher resolution, especially if you don't have a living subject, but you take the brain out of the person, and then you have these kind of custom-made brain containers, which are a bit like the things in Kinder eggs, you know, these yellow boxes that you can click together. The advantage of that is, of course, that in a postmortem brain you don't have any breathing artifacts or any movement artifacts, and the participant doesn't complain if you keep them in the scanner for a really long time. And this enables you to generate MRI scans with much higher resolution. And there were two PhD students at the time, Frank, who was a physicist who basically came up with the scanning routines and did all the measurements, and Arco, who was an engineer who created these specialized radio frequency coils that were meant to be basically as close as possible to the tissue so that we could get the maximum resolution out of that stuff. And then they acquired this kind of data, and I think this is still today the best data set that we have. So this is a whole human brain that was imaged in the 9.4 Tesla scanner at 75 micrometer resolution. And if you zoom in, and I'm not sure how this translates to the screen, I hope you can actually see that. But if you look very closely, there are these two darker lines in the cortex, which you might be able to make out, which we are very, very sure are these stripes of Bayer. So a very prominent feature of myelo architecture in the cortex. And so that was basically the state when I joined. And of course, now that they generated these really high resolution MRI data sets, they started to see this kind of stuff. And so the idea was that we needed some validation, and that was basically the job for my project to come up with methods there, because now that they have successfully pushed the MRI side in terms of the resolution, they wanted to push microscopy methods on the same tissue in terms of field of view and in terms of the volume that you could image. And this naturally led for us to explore light sheet microscopy and optical tissue clearing, of which, of course, you now know a lot. So luckily, I don't have to explain that a whole lot in detail, but just as one thing that is in hindsight a bit funny and a bit interesting. In the first year when I joined, I went to to a light sheet conference in Sheffield, and a lot of the big clearing people were there. And when we talked to them and explained to them what we wanted to do, that we wanted to clear human brain tissue that was half a centimeter thick, that was the original idea. They were all it's not very enthusiastic about it. That they basically said like, yeah, good luck. That probably won't work. So because at the time, a lot of these big famous clearing techniques had already been published, you know, like Clarity and iDisco and so on, but they didn't work that well on human tissue particularly. And yeah, we can, if there are questions about it and so on, we can go a bit into detail why human brain is particularly annoying very often to clear. But yeah, so that was the background on which I started. And of course, I heard you also got an intro on the clearing itself, so I don't have to explain that. But in principle, any tissue clearing technique works in the same way, actually. So you just need to bleach any absorbing pigments. You need to remove any of these tissue elements that have different optical properties to the protein. So remove the fat, remove the water somehow, and then immerse it in a liquid with the same refractive index, and your tissue will turn transparent. And there are, of course, as you probably know, there are a gazillion different ways how to do this. There are so many different protocols. But basically, if we kind of ignore all the stuff here in the bottom, which is more recent developments, there are two big families, I would say, of clearing protocols. You can either do it with organic solvents, or you can do it in aqueous systems, and both have advantages and disadvantages. And in my PhD particularly, I explored those two. Also, again, for reasons, if there are particular questions about it that I can explain in more detail. And first, I would actually focus on the other one, on the solvent-based one, and not yet on the H fruit, because this is actually the solvent-based one is the one that we use a little bit more in the group, because it has a more general application, whereas the H fruit is more like a very specialized application case. And this leads me to MASH. So this was actually my first paper, this clearing method. And this is also something that you might have noticed if you look into the literature, that for some weird reason, especially in the light sheet and in the tissue clearing community, everything needs to have its own weird abbreviation. So we had to come up with our own. But essentially, MASH is a variety, I would say, of this big DISCO family of clearing protocols. And specifically, it is an adjusted iDisco plus tissue clearing protocol, if that means something for some people. And by now, it has also fanned out into different ones that I will show. But yeah, so initially, the idea was that we wanted to combine the light sheet and the ultra high field MRI. And as a first target, because it was a bit more low-hanging fruit, we actually wanted to look at the cytoarchitecture, so at the cell architecture in the cortex. And for that, we used actually this particular microscope that's also shown here. We used the LaVision Ultra Microscope 2. Actually went to Bielefeld, to the company, to image there. And yeah, some of you might actually know that kind of system, because it's also very widely widely used. And it's one of those systems with what I would say is a classical light sheet geometry. So it excites the tissue from two sides, and the excitation objectives would be hidden here in this black box. Then the gray box would be a sample chamber, and the imaging objective in this case is on the top. But I think many other systems have a similar geometry, just sometimes with the camera horizontally instead of vertically. So I think the Mesospin, for example, works in the same way. I think the Zeiss also has the same setup, just with the camera on the side instead of on the top. Yeah, and then of course we combine this with our tissue clearing stuff. And so basically, one thing that I focused on very early on was trying to find labels that are not antibodies, because antibodies are a big pain in the ass if you want to combine them with tissue clearing. So I actually went into older literature, and what I found is that a lot of these traditional dyes that are used in bright field histology, for example, also have fluorescent properties, and a lot of them were rediscovered as fluorescent stains back in the 80s, when essentially this kind of these tracer technologies came up, you know, like an antrograde and retrograde tracings with fluorescent dyes. And people back then were also looking for fluorescent counterstains. And so in this kind of literature, I found a lot of hints for yeah, useful stainings. And yeah, there are many more, but I use or we use mainly these four, which are Nissl stains in different wavelengths. So you can also combine it with different other markers if you want to use it as a counterstain, or also with a cell nuclei stain with methyl green, which is in the far red range. So it is much more useful than DAPI, because something that you might also already know if you have some experience with light sheet and with tissue clearing, that DAPI is not really very useful, especially if you have very thick samples, because you just cannot image very deep when you excite with UV light. So having a replacement in the far red range is very useful. And one of the big advantages of these, all of these dyes, is that they are very small molecules, so you don't have a lot of penetration issues. You can get them deep into the tissue, and they are also super duper cheap because they are just normal histological dyes. And in that way, yeah, you can literally make buckets of the solution without much cost. Yeah, and then this is what we kind of did. We had whole pieces of occipital lobe, so the part at the very back of your brain where the visual cortex is located. And we used, again, specialized 3D printed containers that just fit the occipital lobes, and specialized RF coils. Did MRI measurements on them with yeah, around about 200 micrometer resolution. And then after that, we blocked out pieces around the calcarine sulcus, so around the primary and the secondary visual cortex. And then we made them transparent, and we imaged them on the LaVision microscope. And this was the first kind of proof of concept. And we were very happy that we could already with the LaVision image through the entire cortical wall. And we were able to delineate these typical cytoarchitectural layers that you can see in the cortex. And then when we were zooming in even more with the microscope, we were able to go down to single cell resolution in principle. And now, of course, the cool thing is that we can relate this in principle all back to the big piece and correlate this with the MRI. So that was the measurements were actually done already in 2017, something like that. So we were very happy back then that we got this far. But as many of you will probably know, that if you manage to get something done, then your supervisor will say, yes, that's great, now let's make it even better. So one thing that we knew from the very start of the project is actually what we really wanted to do was having whole coronal slices of occipital lobe, or potentially in the future even of the entire brain. And that means like very big samples of 8 or 10 centimeters in height, and also several centimeters in width. So that was not so easy to do, and like because there is no standard lab equipment to handle that. And at a conference, I met a Professor Herculano Huzel, who works a lot with also with primate brains and so on, and she actually recommended to just use a normal ham slicer from like as it's also used at home to cut these slices of brain tissue, and this works really well if you embed it to keep it from falling apart. And then I had the other problem that for the tissue clearing itself, there is no real standard lab equipment that can fit these big samples to process it in a high throughput manner. And because this project also fell right into the starting pandemic and I was locked at home a lot and I was bored, I bought a 3D printer and I started experimenting with that and came up with just 3D printed equipment that is made of a plastic that can actually withstand the solvents in the mesh tissue clearing approach, which is also important, that something you need to take into consideration with these solvent-based approaches. Very often they are pretty aggressive, and some of the normal lab equipment is made of plastic that will just melt. So you have to use certain plastics and not others. Yeah, but basically now we are really capable of literally mass producing these samples with the original mesh method and investigate cytoarchitecture at a much larger scale. But now the next problem that you have is now you have these big samples that are cleared and labeled, but you can't image them because in a standard light sheet microscope, such as the LaVision, for example, they just won't fit. And this is why we also built a light sheet microscope that has a little bit different geometry. So as you can see here, both of the objectives are rotated 45 degrees so that they are both on top of the sample. And this frees your lateral dimension. So literally the tissue piece can be almost as large as you want, as long as it fits on the stage, and the stage travel allows you to cover the whole sample. And when I say we, I mean mainly Anna, who was a postdoc, joined at the same time with me, and she was the one who actually built the thing. But with, and then we also did, again, used 3D printing to make custom-made chambers, which could house our big samples, and this is how this would look like. So you have your transparent piece, and then these two objectives directly dip into your clearing solution, your imaging solution. And then you would excite from one side, image from the other side, while the sample moves, or rather the stage moves the sample through the static light sheet. And with that, we were able to actually image these whole coronal slabs. So this is something that we were very happy about, and also something that you always have in microscopy. I think this is a general thing. You always have or with any imaging modality, actually, you will always have a trade-off between the resolution and the field of view, right? So even with light sheet, you might not be able to image the whole piece in the highest resolution, at least not in a feasible time frame. So here we settled for more mesoscopic resolution. So this is imaged at 16 micrometers and downsample to that resolution. But if you, of course, if you want to go higher, you can also image, for example, entire gyri in the human brain with 4 micrometer, or then go to the maximum resolution and then, of course, image yet smaller areas. And we use this maximum resolution also to do as a first test to try to do some cell counting and some layer segmentation in this thing. So the layers were still segmented manually, but the cell counting, of course, not, because we are not crazy and that's just too much. So in this first test, we actually used also Arrivis for the cell segmentation, and this worked, I would say, reasonably well, but we were also experiencing some issues with that. So by now, one of our PhDs, he has put a lot of effort in deep learning-based cell segmentation approaches such as StarDist. He also used CellPose by now. And with that, we are actually a bit more happy because they can solve more tricky problems, like when you have these pyramidal cells that have these pointy tips, and if the pointy tips, for example, are too close together or are touching, then these, let's say, these simple segmentation techniques, they usually segment this as one big structure. Or if you have the other way around, if you have a big cell but with not completely even illumination, even fluorescence, then it might segment it into different structures. So there were some problems, but this, I think by now we are in a pretty good place there. One other thing that we also came up with is that in the same paper we also got prostate tissue from cancer patients, where they had to take out the entire prostate. We got this from the pathology department because they're very interested in that. But the samples were embedded in paraffin as is standard in pathology, and then we were not sure would mesh even work on these samples, but turns out yes, you just have to get rid of the paraffin and then it works equally well. And so this is one of those prostate samples. It's a whole slice through a prostate. And yeah, we were able to show that you can basically see these different regions, and you can kind of start grading the cancer scores and so on. So the pathology was also very happy with that. And now this is something that luckily I don't have to do, because now we are actually finally in this position that we have all this MRI data of several occipital lobes, and then we have the microscopy data of these consecutive sections that were imaged. And now basically Johannes is currently working on putting everything together, correlating these slices with the MRI data, which is not trivial. And so I'm very happy that that he's doing that and I don't have to. Another project which is kind of fun, which started more by coincidence, is I'm currently working together, and actually right now I'm trying to get funding for that with the urology department in our university. And they are very interested to look into spinal cord nuclei that are responsible for bladder control. And spinal cord is, again, it's a very challenging piece because it's so highly myelinated, so it's very difficult to clear. And so also there we didn't know would mesh work, and we just threw it as it is on a cross section of the spinal cord, and it works really well. But if ultimately we want to do larger sections of spinal cord, and for that I would need to adjust the protocol a bit to make the delipidation stronger and so on. And I have some ideas for that, but yeah, that's hopefully that will get funded soon. And then I could tell you more, but it's currently still in planning phase basically. And then the last thing that I want to show for this protocol is that we also extended the labeling. So instead of just labeling the cell bodies, the cytoarchitecture, we also tweaked the protocol quite a bit and combined it with several other protocols to also label the vasculature with lectins, so that we have the combined angio and cytoarchitecture. And this project was done in collaboration with the Helmchen Lab in Zurich, because they have this nice kind of open source version of the LaVision microscope, the Mesospin. We went to their place to image our samples there, and we were also very happy with that. And then we could, because we have both the cyto and the vessel architecture, we could correlate those two and then see, for example, how they relate with one another, show that it's actually not as easy as you might intuitively think, that the vessel and the cell densities are not super closely correlated, that they differ a lot also between areas and so on. So, and we think that this kind of information is actually very, very useful for the non-invasive brain imaging crowd. So people that are doing techniques like fMRI and fNIRS, because they typically assume that the microvasculature in the brain is actually very homogeneous, but it's really not. And yeah, so we are curious how they will like this information. So here, this was done with the help of Johannes again. And then we also color-coded these vessels for orientation and did some orientation analysis. And I think already here, depending on how the video streams and translates and so on, you might be able to already just macroscopically appreciate that V1 and V2 look quite different. And indeed, we were able to show, with again the help of some colleagues from our group, that the orientation profiles are also really different, both if you look over cortical depth and as well if you compare different areas. And we segmented layers based on the layering scheme by old anatomist called Duras-Noir, who was an early pioneer in this kind of angio architecture. But we also compared it when we segmented into supragranular, granular, and infragranular layers based on the cytoarchitecture. And yeah, in both cases, we can really see, yes, this is the orientation profiles differ. quite a bit, and we think this has, again, important consequences for things like fMRI imaging, especially if you go to very high resolution and if you want to also delineate layers with fMRI. Okay, so that was all the stuff for the solvent-based tissue clearing, and now, of course, as promised, I will also talk a bit about the HFOO, the Aqueous protocol, and that protocol actually started before my PhD even. I started this together with Professor Ralf Galuske in Germany during my master. And the idea here is that Ralf is really good at this particular staining technique where you have a lipophilic dye, a carbocyanine dye, where you use a crystal of this dye and you kind of poke it into the cortex of very fresh human brain tissue. And then what will happen is that all the cells that are connected with this injection site will take up the dye and become retrogradely labeled. And the idea was, of course, can we make this transparent and then combine this with 3D imaging? And back then in 2015, this was just when a paper came out that showed dye-eye compatible clearing, which was called FRUIT. And we were, of course, immediately jumping on that and we tried to use it. But as you can see here in these 3 mm blocks of mouse brain tissue, it kind of works, but it doesn't work super well, at least in our hands, it did not. So I ended up tweaking it quite a bit to increase the transparency of the tissue. And we were very happy when we then translated this to human tissue. As here, this is a piece of human amygdala that is several millimeters thick. And it still cleared reasonably well. Of course, as you can see, the white matter stays opaque, but that's kind of expected because you leave the lipids intact. And that's strictly necessary because, again, the dye that you have here is a lipophilic dye, so it will get removed if you also remove the lipids. Yeah, this is kind of a trade-off that you have. Yeah, and then again, we imaged it on this kind of weird setup with the two objectives on top. So the sample will always look like it's moving. So that's something that you have to get a bit used to and that you have to remove in post-processing. But again, as a first proof of principle, we were very happy that we could even do it. We could do it in human tissue. And although it took quite a while, it took several years for a follow-up, now we finally are ready to do a follow-up on a study that was actually done even before I joined the group here in Maastricht, also together with Ralf and my current group. So there they used blocks of tissue, the same postmortem tissue that, again, they did MRI on first, and specifically diffusion MRI, where you can reconstruct these myelinated tracts. And then on the same tissue, they did standard 2D sections back in the days and then imaged the dye-eye label to generate ground truth data for the tracing algorithm. And they used this to validate the tracing algorithm. And of course, now the idea is to basically do a follow-up of that in 3D. And I got a whole bunch of samples from Ralf that kind of look like this. So they are also really big pieces, chunks of human tissue. This is all temporal lobe. And you can see here, these dots are the injection sites of this crystal. Yeah, of course, now the idea is to do the same kind of the diffusion MRI, let's say, that we did also for the occipital lobes in the study that I showed earlier, but on these pieces, and then also do the tissue clearing. But because these samples are crazy old, they have been lying around in Ralf's lab for almost 20 years now. So we were not sure how the tissue quality looks like, if the label is still preserved, if they would still clear, because they are like in fixative for a really long time now. So I actually just recently finished clearing or testing clearing for the first batch, and that's how this looks like. So these are, in this case, only 1 millimeter thick sections, and here you can see the same injection site that you also can see over here. Yeah, again, I was pretty happy with that. That even after such a long time, the sections clear basically as well as you can expect. Especially the gray matter gets super transparent. The white matter, of course, not as much, but again, that's kind of the trade-off that you have with this particular technique. And now we are currently debating the best way to acquire the diffusion MRI data, because of course, in the meantime, also the diffusion MRI protocols for these kind of tissue pieces has improved a lot in our group. So now we are putting it all together.
I have a question.
Yes, yes.
What about the blood in the tissue? Because the brain tissue post-mortem, you don't do the perfusion, right?
Yes.
I mean, you just so the clearing procedure, it helps, even though there are leftovers of blood in the vessels, it helps anyway to get the tissue cleared so you can visualize.
Yeah, so that's a good question.
So you do the perfusion before, right?
Yeah, no. So in this case, they are to explain a bit where these samples come from, these human samples. So one of the tricky things with these dye injections, at least in human tissue, with other animals, it's apparently less of a problem. You need very specific conditions, like the tissue has to be super fresh, and ideally it needs to be fixed with a special fixative, like not with normal formalin or something like that, because then with normal fixative, the dye doesn't diffuse so well. So if you want to have good diffusion, you need a fixative that preserves the membranes a bit better. And so basically what Ralph did many years ago when he acquired these samples, he worked together with the forensic medicine department, because in Germany there is a rule: if you suspect that someone was either killed or committed suicide, they need to do an autopsy on the person. And there's kind of a law that allows you to get tissue samples for research if it can be ruled out that this particular organ was connected with the cause of death. And then, yeah, so that means that we acquired these samples at the autopsy, and they were indeed then just taken out and immersion fixed, because there is no possibility to do perfusion on those samples. On the other samples that I showed with the mesh tissue clearing, they are actually perfused, because there we get them from the anatomy department here in Maastricht. And we are very lucky that we are connected to a university clinic as well. So there are medical students that do the anatomy course on body donors, and we basically get a leftover from the body donors. And in this case, they are actually perfused. But if I go a bit back in the presentation, yeah, so if you look at the mouse brain very carefully, I'm not sure if you can actually see this, but you might be able to see that there are some red things still in there. So indeed, the blood is still in there in principle. It's just in the I'm not sure exactly what happened in the human brain samples, why you don't see it there so much. I saw it in some pieces, especially if you have very big blood vessels where a lot of the blood coagulated. But maybe also just because I mean, again, they were washed in fixative for 20 years now. Maybe over time they simply washed out. But yeah, you could, in theory, combine this with bleaching. But again, for this particular use case, that's not really an option, because you already have the dye inside. And if you now would apply bleaching techniques, you would also bleach the dye usually. And so that's a trade-off that you have. So the blood is still in there, and it will give you some background. So you can actually, like especially the erythrocytes are very autofluorescent, but the dye is usually very bright, and there it's not so much of a problem. And also another follow-up that we are currently actually doing together with a joint master student. So one of the follow-ups is the big study with the temporal lobe samples that I just showed, and then we have another one on pig brain tissue, because the nice thing with this dye technique is also suitable for, let's say, non-model species, because you just need fresh brain tissue, and with pig, that's very easy to get because you can just go to the butcher and they give you as much brain tissue as you want. And so we are, one of the things that we are trying to test is that there are derivatives of di, which have a much longer excitation and emission wavelength. And this should be very advantageous for deep imaging, and would also avoid the problem of the autofluorescence with the blood vessels and with the blood in the tissue, because yeah, if you shift this more towards near infrared, then this is not a problem anymore. But these injections are traditionally not done with these dyes, so we don't know if they would actually work. So this is something that we're going to test with a master student. Yeah, basically starting next month. Yeah.
Yes, thank you.
Yes. Okay. Any other questions? By the way, I went through this without asking in the middle, but I'm also actually pretty much at the end. So yeah, just as a summary, those are the two techniques that I kind of came up with and that we are using. It's either mesh or mainly mesh actually, and then now I have several follow-up projects with the H-food as well. So yeah, I hope that in 1 or 2 years I can show you more, because I think that this the project with the temporal lobes is quite a big one. We have I think at least 10 of these huge samples that needs to be imaged in the MRI, that needs to be imaged with the microscope, and that will take quite a while. So yeah, maybe in 2 years from now or so I can show more results on that. But yeah, also maybe I've given you some ideas how you can use the light sheet and the tissue clearing for validation of other imaging techniques such as MRI. Yeah, okay, and with that I'm already done, because I thought it might be nice to have some time for discussion if there are questions about the tissue clearing itself, about like the pros and cons of different methods, or you know, if you have questions like, hey, have you used this and this protocol and so on, what are your thoughts on that? So yeah, if you want to just shoot.
Good. Thank you very much. That was also perfectly in time, and you really managed to pack everything in there. That's amazing. I mean, both the clearing part and the science part.
Yeah.
I will now stop the recording so that people also dare to ask questions, and we can.
Oh yeah,
just
that's fine.